computer scientist that got drafted into doing genomics in the mid-90s, and I've been doing metagenomics since, I believe, 2007, when it became clear that there was a need for a tool to help look at metagenomic samples. And out of that grew the MG-Rast work, which is, has gotten to be quite big. And it's an interesting way to start looking at many samples of many scientists from many continents and integrate the data and sort of build new analysis techniques looking at all this data. Metagenomics offers us access to the dark genomic matter, that is, the unculturables. We can only culture between 1 and 10%. People that don't believe in this number don't beat me up, but it's somewhere in that ballpark. And we have never seen anything that isn't in that ballpark. While all our data is heavily biased by the funding agencies, most of our annotations have been paid for by NIH. So we're not seeing things in our data that are interest to environmental researchers, because the people that have done the genome analyses have been motivated to find things of interest to clinical researchers. And the dark genomic matter really are all the things that we haven't seen before, and there's many enzymes out there that do new and interesting things. Really, what metagenomics is sort of unleashing genomics from the confines of just working in the culturables. This is what is the most interesting thing about genomics to me. Funding, the ultimate problem. Funding in genomics is an interesting game. Funding has been very much center heavy, so they tended to fund the data generators, and I think what's happening is a paradigm shift where we are moving to analysis centers, so people that can actually make sense of the data. But again, I think what needs to happen is that actual scientists that have real ecological questions need to be funded together with the analysis centers in order to make sense of the data. We have to move away from the center heavy funding to go to a different mode. Challenges are that we have a lot of data. We are not the physicist community, that is, we're not very well organized to move data around. So moving data is a big problem, and even if you have the ability to move data, it's kind of hard to come up with a computational means to do the analysis. So we as a community have to start to learn how to share computational results. Even if I have a terabyte of data or terabase of data, interestingly enough, a terabyte of data only has 250 gigabases in it. We use a lot of bandwidth to transport other than interesting data. But even if we have that data, we cannot really afford the analysis. So we have to build mechanisms where I can share analysis results with somebody like Nikos Krividis from JGI, so the community as a whole only invests in the analysis once. Then everybody can sort of share a data product and do the downstream analysis. That is very hard to do at this point, because the standards are emerging as part of the Genome Standards Consortium's M5 project. But what's also happening at the same time, the method development is going on so rapidly that everything keeps changing all the time. And at the funding level that everybody's operating right now, this is sort of an afterthought. The main funding stream is still directed towards the sequencing centers, and that has really to be changed. We have to enable the analysis centers. And just consider something like, I don't know, the prediction of protein coding fragments. It turns out it's in the US, it's very hard to get funding from the people that really write the tools that work well in this area. This is not me, by the way, we're just consumers here. But I'm constantly surprised by the unwillingness of review panels and funding agencies to really fund the things that matter to many, many scientists. I think first of all, what needs to happen right now, people need to be made aware of the fact that all the tools we employ in metagenomics are very, very young. Nothing is solid. Most of the tools we use have been built for the area of genomics, in the age of genomics. So there's the assumption everything is clonal, everything is single cell. And that's an assumption that's totally not true for the environment. So working in a complex mix of pangenomes is really what we need to work in. And I don't know of any single tool that is adequate at this point in time for that. It turns out that if you think of a pangenome, we don't even have a good definition. And more interestingly, we don't even have a single format that would allow me to deposit a metagenome that contains a pangenome, or just one pangenome in the database. There is no tools, there is no format. We just now realize the world isn't clonal, and that's tough.